don't want that. Good morning, everybody. Thank you for joining us today for the thesis defense of Roy Roberts. You mute the speakers on my laptop here, which is logged into the Zoom. Roy is a master's student here at TAMU CC. He started at Del Mar as an undergraduate and then transferred to TAMU CC, where he worked in the labs of Chris Patrick and Simon Geist before I discovered him in my computational biology class and decided this was the student that I was looking for. And since then, Roy has come on board as a master's student. Kind of in the meantime there, he helped with the COVID modeling response. He worked in the lab on barcoding fish from the BioBlitz back in, was it 2018, 2019? A while ago. And since then, he's become an amazing student. He's the only student I've ever had that I've nominated for an Outstanding Islander Graduate Award. I don't know if he's been awarded it or not, but they did tell me to make sure that he registers to graduate. So maybe that's a little foreshadowing. That's what I'm hoping for. Why do I think Roy's an outstanding Islander? Well, he has a perfect GPA. He won a Best Presentation Award at the American Fisheries Society Southern Division meeting in 2023. He's the president of our TAMU CC SACNAS chapter, and really last year as the treasurer, or what were you last year?
It was treasurer.
Treasurer. He helped resurrect the chapter, and this year he led 14 events, 14 outreach events on campus. The one that I like the best is the bringing the National Diversity in STEM Conference to TAMU CC. This conference has 5,500 attendees. It's heavily recruited by every big university and governmental agency in the United States. And we brought all those opportunities back to TAMU CC and had sort of a opportunity buffet for TAMU CC undergraduates. Since 2013, I've been asking the president of the SACNAS chapter to do this. Roy is the only president that's ever made it happen. I asked them to do it, but I don't do it for them, right? So that shows Roy's motivation. I didn't even ask him to do it this year. He just knows I asked in other years, and he made it happen this year. Beyond that, he's earned a lot of awards for money. He's got the Provost Travel Award, Fisheries and Mariculture Travel Award, and nationally competitive SACNAS Chapter Officer Leadership October Retreat Award, which spells color somehow. And overall, I think Roy has been an asset to my laboratory and to TAMU CC as a whole, and he is an outstanding Islander, at least in my mind, hopefully in the university's mind also. So I know Roy has a lot to talk about, so I will let him take it over. Roy, and it's going to tell you about the successful whole genome sequencing of century-old ethanol-preserved Philippine reef fishes.
Thank you for the introduction, and thank you all for coming. I'm going to go ahead and get started. Erosion of genetic diversity, especially in relation to human impacts in marine systems, has increased over time. Populations and systems are continually threatened by habitat destruction, by increasing fishing pressure, and climate change. These lead to events such as coral bleaching, fishery stock collapse. However, with the advent of DNA sequencing, improvements to conservation efforts have continued to improve. With the increasing computational developments as well as sequencing methodology, the sequencing of whole genomes for non-model organisms has become an obtainable reality. With these, we are able to look at changes in individuals and populations spatially and temporally, and metrics such as estimate effective population size, inbreeding, coefficients, and genetic drift can be estimated. Models can be estimated over time, but they have their limitations. Through the use of museum specimens, we are able to essentially improve these since they are essentially a snapshot in time. there is less modeling to be done and is a direct comparison. And then more robust temporal analyses can be done through farther back in time through these. They're not perfect though. Museum specimens have very degraded DNA often. Issues like nicks and gaps, deamination of cytosine to uracil, nucleotide substitution, cross-linking, oxidized bases, and block 3' ends make it difficult or impossible to sequence some of them. However, there are preservation methods that help with the integrity of keeping DNA sequenceable. Two of the most common methods for this are desiccation and ethanol fixing. Desiccation is a drying preservation method that is very common in insects, so like pinned moths, butterflies, beetles, and plants, and then taxidermied animals. Whereas ethanol is, a lot of the genetic work is mainly in invertebrates. Both of these have their advantages and disadvantages though. With desiccation, you have issues with hydrolysis in areas where there is like high humidity, or in situations where the samples are not preserved in cool and dry environments. With ethanol, the concentration of the ethanol degrades over time, and so the concentration of the ethanol to water changes so that there's more water than ethanol, which also leads to hydrolysis and degradation of DNA. There are ways to combat this as well. In a study in 2017 by Sproul and Madison, they used a DNA repair mix to help improve the quality of some desiccated museum specimens that were beetles. They were between 58 and 150 years old in sequencing. And they found that between kits that did use it and didn't use it, the kits that used the repair mix had on average a 30 to 40% increase and got as high as a 60 to 75% increase in DNA yields from these. This kit, what it does is essentially it combats a lot of the issues that I spoke about a second ago. It helps with nicks and gaps, deamination of cytosine to uracil, oxidized bases, and block 3' end to repair the DNA to a more normal state. There are other kits, such as a whole genome amplification. In another desiccated insect study, they used whole genome amplification to amplify low quantities of DNA, so 1.5 and 2 nanograms of template DNA, to create ddRED libraries. They, in this study, did successfully produce high quality libraries. However, there's some issues with WGA that can occur. There are a lot of advantages. It salvages low quantities of DNA. It is an unbiased amplification with high fidelity, so it has a very low error rate in the amplification, and it can amplify DNA between crosslinks, which is not easy to do without. Disadvantages are it increases the potential of contamination. When you have degraded samples, the samples themselves have a smaller size of the genome within the extract. If exogenous DNA gets into this, this will also be amplified with a kit, and likely more so than the degraded DNA. These kits are also costly, and there is precedent for overrepresentation of select sequences based on what is surviving within the degraded DNA. There's very little research done on fish. The two studies that I just talked about were both desiccated insects. For vertebrates in general, a lot of the studies are apes and mammals, but more so invertebrates just in general. Museum fish specimens are often, when sequenced, are single-stranded or metabarcoding projects. There's none on whole genome sequencing, and then there is also no research for ethanol-preserved fish specimens at all. So we're looking at a collection that was collected between 1907 and 1909 in the Philippines aboard the research vessel Albatross. This collection is huge. It contains roughly 17,000 fish with about 2,400 different species. It's in a very unique situation in that it has been exclusively preserved in ethanol since it was collected. It was taken prior to like the fisheries industry booming. and prior to extreme decline in marine habitats as well. So it's a very good comparison for now to the past. So what we aim to do with this study, or what we aimed to do, was to sequence the genomes of senatorial fish and produce libraries that were of high enough quality for downstream population genetic analyses. To do this, we are using comparisons between collection era, whole genome amplification, quantities of DNA, and the use of a repair mix to see if there's a significant effect on the quality and quantity of DNA sequenced. So for the methods, I'll go through the specimen sample collection, the lab methods and sequencing that we used, the experimental design, the informatic processing, and as well as my statistics that I did for this. So these fish came from the Philippines. They started around here. Research vessel Albatross went all the way through around the Philippines, collecting. These stars represent my sample locations for the species that I've used. They were originally stored in high-proof rum distillates, and then eventually transferred to the Smithsonian, where they were kept in 70% ethanol from there forward. These are lots that are rented out at Old Dominion University. At this point, we selected 6 species that we aimed to sequence for this project. Ambassador Ruritania, Iliquidus leuciscus, Doboathurena duodecimalis, Spirania, obtusata, Signathus spinus, and Hypoathurenia temeniki. For our contemporaries, we aimed to find the same species. We went to fish markets to collect different identical contemporary comparisons. At the fish markets, these were field identified and then preserved in 100% ethanol and then shipped to Old Dominion University. So at this point, the species are at Norfolk, Virginia, and Old Dominion University, where they are identified again. They are DNA extracted, QA/QC'd, and then sent to TMUCC. At this point, we do the library preps, the whole genome amplification, the repair mix, and then more QA/QC for the libraries. At 2 steps, we take quantities of DNA after the whole genome amplification reaction and after the library prep to determine how much DNA did the reactions produce, as well as a fragment analysis after library prep to determine the size of the fragments of DNA. After this, they are 150 base pair Illumina shotgun sequenced on a Nova C6000. And it leads us to our experimental design. So for this, the goal was to sequence 96 historical specimens across 6 different species, the 6 that I mentioned earlier. Half of these would be whole genome amplified, half of these would be not, and then half of those would be repaired and half of them would not. So we have 4 combinations of these treatments per. So both, one, the other, and neither. Within these, we are testing different amounts of template DNA with 2 different size ranges. The WGA, since it is designed to amplify small amounts of DNA, is using between 0.1 and 3.2, and no WGA between 1 and 32 nanograms. We aim to do this for both. This was our intention. This isn't what happened. We had several mishaps with species identification, as well as a mislabeling issue that gave us new species that were not intended to be a part of the experimental design. First, our ELE comparison was re-identified as a different species with the same genus. A labeling issue caused some problems with some species that weren't intended to be a part of the project as well. Half of the HTEs were AUR, but those were also identified as a different species later. And so a new species was added. The labeling issue also affected all of our SSPs, which are a new species that also was not intended to be in this project. And then our SOPs would be SSP and HTE. Recently, the DDUs were also re-identified as 2 different species between contemporary and historical. So this is what the overall experimental design ended up being. Like with degraded DNA, we didn't get all the DNA that we needed to for all of the treatment combinations. So these are the values that they ended up being for some of them. And some of them we did not get enough at all, and so they weren't done. So they were removed from the experiment. After sequences come back, a pipeline was developed to quality check the sequences and to process them using FastQC to generate descriptive metrics, FastP to trim and filter, Clumpify to deduplicate, FastQScreen to remove any exogenous reads that are detected in the library, and Repair to repair the paired in reads. Next, we also de novo genome assembly with the short read assembler spades to develop reference genomes for mapping. These are also QC'd with Quast, Busco, Jellyfish, and GenomeScope. For species that we have an error comparison, we mapped using Dodocin HPC and then generated statistics on the BAM files that were produced with samples. For statistics, I am using this general model for most of the tests. For each response variable, we are looking at whether there is a significant difference between ERA, WGA, repair, nanograms per template DNA, as well as the interaction between the four, and then adjusting for species as a random factor. For our normal distributions, I am using the command LME because it has inherent functionality for describing heteroscedasticity structure within predictor groups. Essentially, this means that variance is able to be calculated differently between predictor groups that are specified. I have a few binomial distributions as well. This is being done with GLMER. And for each of these, in situations where we deemed it necessary, we would log transform them with this equation. For all of the models that I make for each of these tests, the best model is chosen through AIC and BIC scores, as well as likelihood ratio tests. And models are reduced based on lack of significance and an improvement of the model with these scores. For generating the significance for each of these, when a model requires, or the model contains a predictor that is a continuous variable, the estimated marginal means and 95% confidence intervals are generated with ggpredict, and then p-values are generated with stat summary. For models that only contain categorical predictors, estimated marginal means and confidence intervals are calculated with e-means. Additionally, significant groups are calculated with CLP and p-values with Carrinova. Okay, so results. I'm going to go through DNA yield from both WGA and library prep, insert length, sequencing success, sequences produced, duplication, and then mapping data for our libraries. So to orient you to this first plot, I have on the y-axis the DNA yield from WGA. This value in the model was log transformed, as indicated by y prime here, and then back transformed for this figure. The figure is paneled by ERA to show difference comparison, and the x-axis is the mass of purified DNA that was added to the WGA reaction as a categorical variable. The dashed horizontal line is added for scale. These dashed horizontal lines fall at 25 on both plots. So everything below this one, this line falls below this line as well. Basically what we found was that WGA did not work very well with the historical samples. They produced far less DNA. In the contemporaries, we found that there was an increase in DNA yield from the WGA reaction when we increased DNA added to the reaction. However, this was not the case with the historicals. We actually see a decrease between 0.1 and 0.32, and then really no difference between the rest. Looking at total DNA yield after library prep, this one is also log transformed, but not back transformed. To interpret log scales, a rule, or the translation is at 0, the base units would be 1. At 1, the back transformed units would be 10. at 2, 100, at 3, 1,000, just for scale. We found in this that higher yield with more DNA in library preps for most of our treatment combinations. We see that the grouping of contemporary had an increasing DNA yield, and that the differences between the slopes of the contemporary no WGA and then both of the historicals was not significant. They essentially had the same trend in increase. This was not the case for contemporary WGAs, as there was no evidence of an increase in DNA yield from an increase in input DNA. Additionally, the dashed horizontal line here that falls at 2 log units, or 100 nanograms of DNA, was our target for the amount of DNA that we wanted to get from this reaction. For each of these, we wanted to sequence 100 nanograms of DNA to have an equal amount of sequencing effort. However, this was not possible in most of our historical samples. So we would sequence the ones that, the maximum amount that we could for those. So moving on, this plot is essentially the same. The difference is the y-axis is now mean target insert length. Here we see that DNA fragment length is indistinguishable between the WGA treatments. So between ERA, the contemporary and the historicals are pretty much the same, like they're statistically identical, which is not the case for no WGA. We have significantly less or significantly smaller fragments in the historical no WGA than either the contemporary no WGA or any of the WGA treatments. Additionally, for this, the horizontal line falls at 300, in this case, 300 base pairs, which is the maximum amount of sequence that we get from each of the reads, right? We're doing 150 base pair paired end Illumina sequencing. So the sequences on either side, 150 in, are sequenced, and the middle portion of the insert is not sequenced. So inserts that have less than 300 base pairs will have overlap in what is sequenced. And so that is what we're seeing with these historicals, and not with either of most of the libraries for these. So looking at the success of these libraries, the main takeaway is that WGA hampered success in historical samples. A significant amount of libraries failed in the post WGA, and the no WGA succeeded far more often. Most of our contemporaries succeeded. There was one individual in the no WGAs that failed, and then like one library in the plus WGA that failed. But for the most part, they were fine. The big takeaway is that the plus WGA failed quite a lot. Looking at the libraries that failed in relation to the massive template DNA that was added, we see that there is an inverse relationship between the two WGA treatments and success and mass of input DNA. For the no WGA samples, as we increase in DNA added to the reaction, the probability of success increased. In the WGA, as we increased in DNA, the probability of success decreased. So more DNA caused them to fail more often. This is likely due to the WGA reaction requiring the Phi-29 polymerase, and is likely impacted by enzymatic inhibitors that are inherent to the albatross collection, the preservation solution of them. So in looking at the amount of sequences that we got back, these are normalized by the amount of sequencing effort that we did. Looking at the raw read to be an unfair comparison, since we had different amounts of DNA sequenced per library. So these are adjusted for that specifically. We had the fewest reads per sequencing effort in the historical no WGAs. They are significantly less than either of the other treatment combinations, which all statistically group together. The contemporary and plus WGA libraries produced similar results, and saw no discernible effect of template DNA on the amount of sequences that we got back from sequencing. We saw very high levels of duplication. This figure has duplication on the y-axis and then mass of initial template DNA on the x-axis. They are scaled differently so that we can better see the no WGA. These are also log transformed values that have been back transformed. But for the big takeaway, no WGA has very, very high levels of duplication. So what we're seeing is overrepresentation of specific loci. For this, the duplication in every single one of the treatment combinations has a decrease in log units as we increase in template DNA. So more DNA meant more less duplication, I should say. Looking at our mapping results, the genome mapping coverage and depth increased with more reads in historical libraries, but this was in historical minus WGA libraries, but not in whole genome amplified historical libraries. So looking at specifically these, we see that there is a positive correlation, a very strong linear relationship with the amount of coverage of the genome that we sequenced for historical minus WGA libraries. We also see that in depth of coverage increases linearly as we increase in sequencing effort for these libraries. Unfortunately, we only had one species comparison for the mapping result. AUR is the only one that we had both a contemporary and a historical. And so that's the only data that is here. For these, we assembled de novo a genome from the libraries by concatenating each of the no WGA contemporary libraries to increase like sequencing effort, I guess. For the WGA historical libraries, we see that even though there's very high sequencing effort here, there's a lot of reads between like 22 and 25 million reads in these libraries. There's very, very low coverage of the genome, but with the depth of coverage is very high, between like 400 and 700 in these libraries, despite such a low percent of the genome covered. So going into the discussion, the things that I want to discuss are the effects of the imbalanced design, whether or not we were successful in sequencing the genomes of the historical specimens, the enzymatic inhibitors that are found in the historical samples, an assessment of WGA, and the effectiveness of repair, and then the use of more DNA in library prep. So I've had two general reactions when talking about the species issues, and one of them is usually a positive reaction. More species is better, and one of them is a question as to whether or not this kind of invalidates the experimental design, as it seems that we may not be able to adjust for or control variance between era by having different species. However, this is unlikely to be the case. In this figure here, we see the ones highlighted are all from a single species, right? The results between the contemporary and the historical are significantly different. And within era, we see that they essentially behave very similarly in how they resulted. These all have a general positive increase over time, and these are all mostly flat and randomly dispersed here. Additionally, we do control for species within our statistical model anyways. It is factored in as a random factor. And so when variance is calculated within the model, it is already adjusting for species. And if species were the leading cause for variance, then repair would no longer be a significant factor in the model. So this does have benefits, right? So more species, more results for more species, which means there's more support that this works across different species. This methodology works across different species. However, because we don't have the species comparison, we have fewer mapping results essentially. With more mapping results, it would support some of our claims a little more. We believe it's very unlikely that historical libraries are just contamination from the contemporaries or contamination from something else. There are a lot of precautions taken with these samples in the lab methods as well as in the informatic processing. Laboratory precautions include protocols done on different days, on different plates, with flame sterilization and regular cleanings for lab equipment and lab area. Informatically, we do quite a lot of filtering for our libraries and our genomes. For the libraries themselves, we use FASTQ screen to test against many different databases of nucleotides to remove any potential exogenous reads. For the genome that we are mapping, we have very stringent filters for removing potentially exogenous DNA, removing small scaffolds, removing really high and really low GC content, removing really high depth of coverage sequences, essentially, from these de novo genomes. Additionally, the results also support this claim. We see significant difference between error for pretty much everything that we've done in most of the tests, I should say. Especially in success, if there was continued contamination in many of these, we would likely see similar success between errors, but we don't see that. We also see significantly different sizes of inserts, mean target insert length between the WGA treatments specifically. For this, all of these are well below the ideal mean target insert length that we would be looking for. And all of these are above. If there was contamination, I don't think that this difference would be nearly as big as it is. Additionally, there are just generally very few opportunities for contamination to occur in a lot of our processing and our lab work and support. If anything is a takeaway, I think it's that WGA did not work at all. It was detrimental to the sequencing of a lot of these fish. There is a very low probability of success even between the historical treatments. We saw very poor amplification of DNA yields after library prep, as well as amplification of DNA from the WGA reaction. A lot of the WGA reactions produced values that were similar to what we would either be starting with for the library prep or less in some cases. I guess the one strange result that we have was that the insert lengths for the WGA libraries were almost identical to the contemporary. This might be some form of contamination. I think it's unlikely that it is fish contamination based on what I stated previously. It might be some kind of bacterial contamination, but it seems that there's possibility that the WGA reaction just amplified larger portions of the degraded DNA, I guess, in the sample. Additionally, we kind of expected the best treatment combination to be the both the repair mix and the WGA. The repair mix ideally would have fixed any damages with the DNA, creating longer fragments of DNA, and then the repair mix or the WGA would then amplify those larger fragments. However, this was not the case. There was no synergistic effect between the kits at all, which is another failure of this method. There was very high levels of duplication in these samples, like an absurd amount of duplication for having gone through all the filtering that we've done. This duplication essentially indicates that while they're not like completely identical sequences, as we remove perfectly identical sequences, at least parts of them, enough of parts of these sequences are exactly the same. And we see that with the depth of coverage. The low genome coverage with very large sequencing effort in AUR, we see huge depth of coverage for very small amounts of the genome actually sequenced. Within the WGA reaction itself, there is also inherent enzymatic inhibitors. In French 2021, they found that the DNA polymerase and restriction enzymes were inhibited in DDRAD sequencing of these same specimens. Additionally, alcohol, like sugarcane spirits and other alcoholic beverages, are known to have impurities in them, and even metals in some cases, especially in alcohol that is produced with like low quality water. For example, alcohol that was probably made in 1906 didn't have the best water, and likely had some kind of contaminant or metals in them, which is likely leading to the enzyme inhibitors. We see that specifically with the probability of success. As we increase in DNA, so as we increase in the amount of the product that is added to the WGA reaction, there is an increase in chance of failure, which is not the case in the no WGA. So then why does W minus WGA perform better than the whole genome amplify samples? So our protocol itself has a very low dependency on enzymatic function. The very first step of the shotgun sequencing is an enzymatic digestion. However, if this is inhibited, it likely doesn't have a huge impact, as the DNA is already very fragmented and very degraded. So it's almost as if it's already gone through the process of the enzymatic digestion, if it is being inhibited. Additionally, the enzyme that is used, like the ligase, does not seem to be nearly as susceptible to the inhibitors within the preservation, as Martin found out previously. PCR and DNA polymerase does not have to work for this library prep to be successful. And so it is not reliant on these specific enzymes. Every reaction that we do within these also comes with a bead cleanup, or a paramagnetic bead cleanup that will remove contaminants, excess proteins, bad sequences, low quality DNA from these. And so as the reaction progresses, we have likely less and less of these enzymatic inhibitors, which means that at the point when it might matter during the ligase, there's likely less of these inhibitors to cause problems with the reaction. This overall study was entirely inspired by French's discovery that the inhibitors caused almost all of the DDRAD sequencing libraries to fail. The shotgun sequencing protocol itself was the simplest and least dependent on enzymatic activity that is available while still using commercial kits. And so this was the method that we went after Martin's study. Looking at repair, A lot of studies have found that repair works very well. The Beetle paper that I referenced previously found huge success with the repair mix, 70% increase in DNA from just fixing some of the DNA is a huge amount. However, we did not see that. There are two propositions for this that we have. Either there's not any of this damage present in the degraded DNA that we have, so there's nothing for it to fix. There would be no evidence of nicks, blocked 3' ends, oxidized bases, deamination, cross-linking that the repair mix would have fixed. Additionally, it could be that the enzymatic inhibitors that are within the ethanol might be inhibiting enzymatic digestion of the specimens over time anyways, and so it might be a boon rather than detrimental to the preservation of these species. Alternatively, it could be that the repair mix itself is completely inhibited by the inhibitors, and it's just not working. It's not designed to amplify DNA the way that WGA is, and so the repair mix itself failing would not destroy or we wouldn't lose nearly as much DNA like we would with the WGA reaction failing. So for this method, we have determined that this is a suitable method for generating population genetic analyses. Based on our coverage data, we can see that there is a general increase with sequencing effort. As we add more sequencing effort, right? So these are our speculative extrapolations from the relationships that we see. We can get somewhat close to how the contemporary samples get with coverage. This is unlikely to be exactly how it is, but this relationship here is strong enough that is likely going to approach the asymptote of how much coverage we would get with the degraded samples with more sequencing effort. Additionally, with depth of coverage, we see a very strong linear relationship with an increase in sequencing effort. So more sequencing effort would mean more coverage and more depth of coverage. There are also other methodologies that are suitable for this that are very similar. Low coverage whole genome sequencing as well as target capture sequencing work very well with this, and mostly build up the same principle. There are limitations with this study. For example, we lack the ability to map all the species that we had due to a lack of genome availability, due to taxonomic misidentification, as well as the labeling error that was described previously. We only evaluated 10 out of the almost 24,000 species of fish that were within the albatross collection, which is a very small subset. We only evaluated specimens from the albatross collection. In a different collection, it could be that there are different results, but only looking at this collection is a limitation of the study. We had limits to DNA extraction values. The DNA that we extracted was approaching, did not reach a lot of the values that we needed to, which is understandable. It is very degraded DNA. And so it's difficult to get the amount that we wanted. We can't technically be 100% sure that the enzymatic inhibitors exist, as we did not test for them. However, there is very strong evidence to imply that they are there and causing issues with the reactions. It was difficult to obtain fresh specimens from the markets, which also likely led to the reason why we didn't get enough DNA in some of the contemporary samples. At the markets, the fish are kind of out, and so they are sitting in the sun dead, and their DNA is very quickly degrading. So having to go through and get them, and then identify them, store them, ship them, likely caused some kind of issues, which led to some of the low quantities of DNA that we're getting from the extracts. And then last, our sample size was very low within species. We tried to, with our experimental design, use one individual per species when possible. This wasn't always possible. Most of our albatross specimens, we have multiple individuals, but having 1, 2, or 3 individuals doesn't really prove that it is entirely possible to apply this to a population. However, in a current study, we have done this, so it has been shown. In conclusion, it is possible to sequence the genomes of century-old ethanol-preserved fish specimens from the albatross collection. Standardized library preparation protocols using commercially available kits can be done with only minor modifications to them. Whole genome amplification did far more harm than good. The repair mix had little to no discernible effect on any of the metrics shown here. And then using more DNA in library prep yielded far better results than using less. I would like to thank all these people, Chris, Sharon, Eric, especially for always helping me with everything throughout the whole project. Kent, Dr. Alcala, Dr. Malin, Brendan, Abner, Jem, John, Renee, Ivan, the Smithsonian people, Jeff, Diane, Glenn, Jason, Martin, Maddie, Dave, Frauke, David, and Jordan, especially for everything that you guys have done to help. Then everyone in the Pyre that I didn't name individually as well. Everyone's been very supportive and helpful throughout. I would like to thank my family and friends. This project was possible because I would receive funding from the COVID-19 task force to pay my salary and tuition, and then also from the core lab in the second half. And then this project was funded by an NSF grant. Anybody have questions?
Okay, we can take some questions from the room, and I will intersperse questions from Zoom. So, what is it?
Oh, yes.
Knowing now what you know about WGA not being as effective, is there some sort of method or strategy that you would jump into going this fresh? Like, because you mentioned that there's all these enzyme inhibitors. Is there something, would you just like go into the bead extraction of the DNA now, or?
Yeah, I think what would be most effective would be improving DNA extraction technique as opposed to trying other kits within that may also be reliant on enzymes. Just getting more DNA from the extraction seems to be maybe the best thing that we can do at this case with this methodology.
We'll stand closer to the mic just so people on Zoom can hear.
This mic doesn't work. It's these ones up here.
Okay. All right, from Zoom. Roy, would you expect this type of repair to help when performing amplicon-based sequencing of contemporary formalin-fixed samples, paraffin-embedded samples?
The repair mix? No, the repair mix isn't, well, it is for formalin-fixed paraffin-embedded, so it should help. Yeah. Yeah, the answer is yes. I would expect it to help.
Yeah, that's why they named it that. Another question from Zoom. How do these results inform conservation efforts?
So by looking at changes over time and looking at how genetic diversity has changed and how the effects of I guess the anthropogenic effects on these populations have shifted genetic diversity, we can take measures to I guess stop those forces. Yes.
You made two comments that I'm trying to square off a little bit. You mentioned that you looked for exogenous DNA in your libraries. Great idea. Well done. And very nice talk too, by the way. But you also said there might be bacteria or virus contamination in the libraries, which you bioinformatically can look for. So I wasn't quite sure how those two statements squared together.
They are the same.
Okay.
I mean, like our FASTQ screen filters for like 81,000 bacterial genomes and a lot of virus genomes. I don't know the number off the top of my head, but
All of them. But not all of them are sequenced.
So we do remove it with FASTQ screen.
Good question. See, oh, there we go.
You mentioned that the fragment length size was like 300, and it was odd that, you know, it was in the two contemporary and historical. Do you think that has to do with the depth of coverage, or that it was just overrepresented and that 300 was just like some kind of amalgamation of them just over and over of that one region? Or do you think that was just an anomaly?
So for I don't think it was an anomaly. I don't think it was contamination. I think that it could be that the whole genome amplification, which is known for or which is designed to amplify like cross-linked DNA, maybe allowed access to larger portions that were unsequenceable for just the library prep method, which could explain some of all these large fragments. With these, I don't know, larger fragments, then they increase amplification of like specifically those, which might lead to the ridiculous depth of coverage. Yes.
For the ones that you did whole genome amplification for and got real high depth, but on limited areas, were they the same areas between the genome preps, or were they different areas within the genome?
I have not checked.
Okay.
I did this like 4 days ago.
It would be interesting if it was always the same areas that you were able to get, especially if you had different fish.
I will be going through like IGV and looking to see like, you know, what's happening, but even between the historical ones too. But I haven't gotten to it just yet.
Anything else online? I'm doing so many different things. I had a question, but I forgot. We have a couple minutes. Let's see if I can conjure it up again.
So different tags can be used for different things. Like there are some tags that will actually sequence across uracils, while other ones won't. Would you consider using different tag polymerases for different parts of this, or for saving some of these genomes?
With polymerases in general being like known to be inhibited, probably not. When this method is like really simple and generally very cheap, and so increasing cost to try to change the way the sequencing is happening, or to I guess improve things in this manner might be inefficient for cost. When we see that really what improves it is just higher sequencing effort.
Okay, I've got my questions back. Thank you, Andrew. All right, Roy, you said that there wasn't a lot of research in trying to sequence fish museum specimens, but actually there, for 2 decades, between 1980 and 2000, or even 3 decades to 2010, there was a huge amount of effort to PCR amplify like CL1, a mitochondrial gene, and sequence these specific specimens in that way. There's a reason why there's not much in the literature about it, beyond a lot of failures. And there's a reason why we're not sequencing the, you know, paraffin or sorry, the formalin fixed specimens from museums, it's because one, like I did before this study, I thought everything was in formalin, but that's not the case. But the other thing is that it messes up PCR. Why did this work when that didn't work?
PCR is not required for this sequencing methodology, so it's not reliant on the DNA polymerase within PCR. I believe when I said little to no research, I meant specifically whole genome sequencing for museum specimens. And I think I did mention that there was metabarcoding and single-stranded research within museum specimens for museum fish.
And perhaps the 3 decades of failures kind of has deterred people from even trying something like that.
Yeah.
Kind of just assuming that it's not possible, or at least a fool's endeavor. My last question is, and there is a right answer to this, but I want to hear what you have to say about it. If you, now knowing what you know, if you want to improve this and make it better by altering some part of the library preparation protocol, what would you do? What part of it would you want to try to improve upon? I won't tell everybody what the right answer is. We'll find out in the private session.
I guess what had the greatest effect was the cleaning, right? And so an additional cleaning might help increase the amount of DNA that is available.
Every time you clean, do you get more or less?
So you lose some DNA, but if it affects the way the like the kappa kit works, then it might be worth trying.
Okay, what else besides that? Knowing that as you clean more and more, you get less and less DNA, and as you get more DNA, you get more success.
What would that change? Specifically the library prep protocol?
Somewhere, anywhere from tissue to
From tissue
to sequence.
I don't know.
Okay, so final answer is the cleanups.
Final answer, yeah.
I do think more cleanups would help remove contaminants
So
as well as DNA. All right, I don't see any more questions from Zoom, and I think we've exhausted the questions in the room. I think we have one more round of applause for Roy, and then we'll. Moment here in between the public and private session to talk to Roy and tell him what you thought about his talk, and then we'll ask you to leave by, say, 10 minutes from now. Thank you.